So you're currently looking at a whole lot of these different squat lobsters from around the world, is that right?
I'm looking at Australian ones, so I'm looking at describing new species from Australia, but I'm also, to understand what species we have in Australia, I have to look at all the different squat lobsters from all over the world, and there are, of the group I'm working on, which is a type of squat lobster, there are 250 different species around the world, so it's hard work trying to compare the species that we have in Australia with all those other species around the world.
So when you say you're describing them, does that mean that you are kind of identifying them for the first time, or?
Yeah, so we have, when we go and explore the ocean and the deep sea, a lot of the species we find are new, and most of the Australian species of squat lobster, we don't even know what they are, they're new species. So my project is to describe those species. So
how do you start? Do you just look at ones that look a bit similar, or?
Yeah, so I have to, when you make a species description, that describes all the different parts of the squat lobster and how it's different from everything else that's known. So we do illustrations and take photographs and write complicated descriptions about which spines are, how many spines are on the third article of the fourth leg, and all that sort of detail. So that's part of my project.
Fantastic. Now, Anna, you were going to show us how you would describe them by doing a diagram for us.
Yeah, so to do an illustration, it's pretty hard. You can see this is that's a big one, and many of the squat lobsters that I work on are really tiny. They're only a few centimetres in size. So we use the microscope, and I look down the microscope, and at the same time I can trace what I'm seeing under the microscope onto paper. So I thought I'd do a live drawing today of a species that I think is probably new. It's a little species which its genus name is Munida, and I think it's closely related to a species called Munida leogora, which is known from New Caledonia, which is quite far from, we found it in the deep sea off Western Australia. So I'm not sure if it's the same, and I think if I do this drawing, maybe you guys can have a look on and we'll, I'll see if we can compare the two species.
Okay, great.
Cool. Okay.
Let's get started.
Well, I might, I'll put this guy away and I'll head over there.
Fantastic.
Guys.
So we'll check in with you in a minute.
Yes, that sounds good.
Great. All right, so next, Hugh.
Hello.
Welcome. You are looking at deep sea invertebrates as well, aren't you?
I am, yes. So I'm involved with a large project that involves several other different museums and institutions, and we're exploring the diversity in the deep sea, specifically in the Great Australian Bight, which, for those of you who don't know, is that sort of that large curving section of southern Australia, the big open ocean that's out there. And we don't know a whole lot about the deep sea in that area, so we've had the chance to go and do some collecting, and it's up to us now to identify the species and identify all the specimens that are there, and sort of get an idea of how many species are out there.
Yeah. So I've heard, read that we know more about the Moon and Mars than we do about our oceans. Have you guys heard that, and can you comment? Do you think that's fair to say?
I think that gets shown a lot. You know, it gets discussed a lot. And for one thing, it's, you know, we can look at 50% of the Moon with a telescope because it's right there. You know, we can't really look in the deep sea. We have to be, you know, it's a lot of work to actually get some really observation equipment down to the deep sea. So, you know, and people have definitely, I think, spent more money, you know, in the space program and, you know, sending robots to Mars than we have sending robots down to the bottom of our own oceans.
Why is it important to know what's at the bottom of the sea?
Well,
why do we need? Why? I mean, I'm interested. Yeah. You're interested. Di's interested. Anna's interested. Elka's. We're all of you are interested. But I mean, why does it matter?
Well, deep sea biology is really, really interesting because we have a pretty good idea of what biology is like in the rest of the world. You know, you can think of, you know, what the forests or the shallows, But down in the deep oceans, life has evolved to a whole bunch of really interesting conditions. It's really dark, it's really cold, it's a lot of pressure. There's also areas like deep sea vents where it's incredibly hot. So animals have evolved to fit all these different niches, and there's some hypothesizing that some life may even have originated in the conditions like in the deep sea, these sort of deep ocean hot vents. So without understanding what the conditions of life are like all over the world, that gives us much better insights what life or how life originated in the rest of the world, but might even be what are the kind of conditions that life could live in other worlds or other planets. Like people are, in our own solar system, I think some people believe the best candidate for life is one of Jupiter's moons. It's covered in a frozen ocean, but maybe down under the ice in sort of a deep sea environment, there could be hot vents, and so it's things like that. There's an application there that you don't necessarily think of.
And some people have actually suggested that we shouldn't really be called Earth. We shouldn't really be called planet Earth. We should be called planet Water or planet Ocean, because 70% of the surface is ocean.
Yeah.
And the trenches in the deep sea, the Mariana Trench, is deeper than the Himalayas, deeper in the ocean than the Himalayas are above. So it's a huge explored, unknown
It's not just flat, is it?
No.
It's quite
Seriously not.
complicated.
No, there's mountains and huge mountains and trenches down there.
All and so much of it is unexplored.
So to get to the top of the Himalayas, we climb up. How do you get down to the bottom of the oceans? Do we send robots? What do we do?
We do. I mean, there are submarines that people can sort of pilot down there. There's also robotic equipment that can be sort of driven down there remotely. There's now new technology, but like how we have drones in the air, we have drone submarines. So things that don't even have any connection, they can automatically go down and search. But when we're doing biology research, we either usually use a robot, an ROV, to go down and observe and maybe collect. But what we've been doing lately is relying on sort of the old-fashioned methods, which is sort of like fishing a little. We go out in a boat, and you have sort of a net you can drop down, and when the water is 3,000 meters deep, it takes almost 3 hours for the net to hit the bottom. So you sort of have to hold course, go a little slowly, start lowering this net until it hits the bottom, and then you pull it along the bottom for maybe 15, 20 minutes, and then you pull it back up again, and it's another 2 or 3 hours to come back to the surface.
Yep.
And then, only then, when you get it onto the back of the boat, do you know if it actually, did it get a good catch or not? And so you pull everything on board. Do you have the photo up there? So this is us right there. This is one of Australia's other research boats, just pulling up what's called a beam trawl. So we don't know, you can't even see the back of the net yet. You don't know what kind of catch is brought in. Cam, do you want to bring up the other photo there of? So you might bring up the catch, and you sort of put it into a Nelly bin, and then you can find, here's actually what we've caught. But then you've just got a big bucket full of mixed specimens. So you take it into the lab on the ship while everything is still fresh, and then you sort of biologists sort everything out, and you try and preserve things quickly, and to keep the DNA intact. So this is an example of in the lab, having buckets and buckets of material, sort of sorting through to find all the tiny little animals. So it's a very exciting and sort of exhilarating thing, because everything you pick up could be new, especially if it's in the deep sea environment.
Great. So we touched on why it's important for us to know about what's in the oceans, but particularly about the deep sea. Why do we want to find out about these things? We're never going to come across them, so why is it important to us to know about them?
Well, it is important to understand what's out there, because we do have impacts on the marine environment in lots of different ways. And there are things like deep sea activities like deep sea mining or drilling for oil and gas. And all those things can have impacts on the natural environment. And there's large areas of Australia's coastline, like the Great Australian Bight, where we knew almost nothing about the deep sea, at least beyond a few thousand meters. There've been no real sort of major exploration. So this is, in a way, it's really kind of what's out there. We need to know what's there. So are there really rich, species or groups of species out there, is it have a lot of diversity, or is it sort of fairly typical of the rest of the deep sea?
Yeah.
So these are all really important questions you need sort of answered.
Most people don't know that there are deep sea coral reefs, corals that have been growing for thousands of years, individual
Absolutely.
corals that have been growing for thousands of years in the deep sea. Huge branching corals, and they create habitat. Whole communities of invertebrates and fishes live amongst these fantastic coral reefs down in the deep sea, and we need to know about that. We also, Eric's whales, they feed, and many species of fishes, commercially important fishes, are feeding on things in the deep sea. So, you
So
know,
there's a great deep reef.
Yeah.
Yeah.
So it's sort of
GDR.
this thing of, you know, it's out of sight, out of mind. You forget there's all, you know, what's down
Yes.
below in the darkness there. You know, there's a lot of hidden diversity that's there.
Great. So in some ways, knowing what's down there lets us know in the future how things change, but it's also a model for maybe changes that are happening
Absolutely, yeah.
Absolutely.
Yeah. elsewhere. Great. What we might do now is just throw over to Anna and have a look at how her drawing's coming along. So, Anna, correct me if I'm wrong, but what you're looking at is you're looking down through a microscope. One eye is seeing what's in the microscope, and the other eye is seeing the piece of paper that you're drawing on. Am I correct?
I think that's correct. It's funny, when I'm looking down at, I'm used to just seeing it as one thing, but yeah, I think technically one eye is, I can test now. No, I think they're both
So,
at the
but it's a
same
split image. You're sort of seeing both things at the same time. Is that right?
time. Yeah.
Okay.
So it's like having both images. I can see my hand moving here, and I can see little squat lobster there. So I'm pretty much just tracing over the squat lobster.
Yeah.
And not every kind of microscope can do that. It's a special microscope that we have there with
Yes,
that
it's
arm that comes out the side of it is a special adapter that lets you draw, sort of, you know, see what you're doing while you draw and trace something.
Yeah.
So this is the illustration, I don't know if you can, of the one I was telling you about from New Caledonia. And so you can see it's pretty technical. It's a line drawing of the carapace and the tail, all the different body parts that can be used to distinguish this species.
Great. All right, we'll leave you to it, and we'll come back and check in a few minutes. All right, we're going to move over to you, Di. You are the Senior Collections Manager of Vertebrates at the museum. What
Yes.
does that mean?
Well, I'm a fish person. I oversee the vertebrate collections or management of the vertebrate collections, but I'm actually work on fish, an ichthyologist. We have large research collections at the museum, and again, another thing that many people who don't work in museums are unaware of, that we're not just the exhibitions that are on display. The actual heart and soul of the museums are our research collections. Some are on display, but many, many, many more are behind the scenes, and they, apart from supporting exhibitions and education, they support research. They support our knowledge of our biodiversity and how it evolved over time. And without those, I call them they're in the natural history extant collections, the animals that are still living today. Even though they're dead things in jars, I call it a living collection, because certainly a biological library, because they continue to give up their stories over time, and it belongs to the people of Victoria outside. They're all the collections for now and future generations to document what we have in Victoria and the wider Australia and the world, and also how it changes over time.
Yeah, I think the notion of a living collection is really important,
Yeah.
because it's not things that are just hidden
No.
away, but researchers are constantly coming and going. We're getting new things are going out on loan, we're borrowing things, so it's a really very dynamic thing.
Absolutely dynamic, yeah, and yes, I don't think we want it to be the library book sitting on the shelves. We want it to be a working collection.
The museum basement is not covered in dust.
No.
It doesn't settle. There's no time for it to settle.
No, yeah.
So what specimens have you brought in
So-
for us today?
I'm fascinated with deep sea fishes, but I haven't brought those in today. Deep sea fishes are awesome. They've got lights and big teeth in the main. But I brought in a bunch of sharks because our sharks were in the news, I guess last year, when we got a big basking shark, and we have the big basking shark heads still being preserved in a big tank. I've brought in they're the teeth from, and I think there's teeth from a basking shark. I'll keep it still, and they're tiny. I mean, sharks are in the news. Sharks are in the news for a whole host of reasons. A, they sometimes aren't very nice to people, generally white sharks, or I guess the 4 top predators are white sharks and bull sharks and tiger sharks and oceanic whitetips that people don't really come in contact with. But sharks vary from this little fellow here, which is not quite adult, but it's a pygmy lantern shark. This one's actually from Taiwan, but they do occur in Australia. It's bioluminescent, so it creates light along its belly, which serves to camouflage the shark. It lives in the oceanic midwaters. It doesn't live on the bottom, and it doesn't live up near the top where the sun goes. It lives in oceanic midwaters where light gradually filters down, that kind of twilight zone. So little animals in there have got nowhere to hide, so they have to create ways to hide themselves, and many animals do it by having bioluminescence where they light up their bellies, so that any animal looking at them from below just sees they blend in with the kind of lighter sky, and they're always dark on top, so they blend. If you look down on it, they just blend in with the dark sea. But these guys, yeah, they're really cute. Definitely not man-eaters, definitely not scary.
You know that's a shark.
Yeah, how do you know it's a shark?
That doesn't look like a shark.
Definitely doesn't look like a shark. It's got 5 gill slits along the side. Bony fishes have 1, can you get into that? 1 gill opening. This has 5 gill slits along the side. Sharks have between 5 and 7 gill slits along the side. I can't show that. It has a cartilage and a skeleton, and it has the general body proportion of a shark. It has a pointy nose. It has lots and lots of tiny little teeth in the jaws, and there is an image of the teeth in those jaws that I've got, Cam. Sharks can replace, no, not that one, but that shows this. This will do. That shows this. It's a tiny little one sitting on black. That'll do. So they can replace their teeth, and the teeth are kind of like, kind of made of the same stuff as our teeth, as are all the rough bits on the skin. This one's only got 1 dorsal fin. It's got pectoral fins. It's got little pelvic fins back there. I mean, it's a basic shark shape. And it has lots and lots of little sense organs on its snout that you can't see in this. But, I mean, they're the basic characteristics of sharks, which are not primitive animals. They're a highly evolved group of cartilaginous fishes. Sharks and flat sharks, which are rays at all. And sharks are also incredibly important in our biodiversity and in food webs. They're apex predators, and they're seriously in trouble. Not just sharks, but also rays, because of primarily the shark fin trade, which really decimates shark populations, and fishing for food. And why that really affects sharks is that they generally take a long time to mature. They all have internal fertilisation. They produce relatively few young, and so they're slow to build. If the population's collapsed, they take a long time to build their populations up. White sharks might have 1 pup every 2 to 3 years, so they can't build their populations up. And it's thought that white sharks may be becoming more common, but more people are in the water, so there's a bit of a debate about why white sharks are interacting more than people. And white sharks are one of the few sharks that they feed primarily on seals or passing whales, which is why they're often around during winter, whale carcasses. They can't immobilise a big seal in one hit, so they come and do a mug shot, and then they'll go back and feed. And so people generally don't see white sharks, whereas other sharks will come and have a pier. A very big white shark. And Cam, can you there's a really interesting story of white sharks in Port Phillip Bay. There's a story there. So can you get the writing bit up? Yeah, let's see if we can see that. I don't know if you can read that, and I don't have a copy here, but in the 1870s, there was white sharks, sorry, I've dropped the microphone thing. White sharks,
It wasn't a shark attack.
no, it wasn't a shark attack. White sharks attacked, in fact, I think prior to about 1930, there'd only been a couple of reported white shark attacks in Port Phillip Bay. So white sharks did occur in the bay, but not very often. And they still occasionally occur in the bay. But this is one particular one where there were baths, Brighton baths, and I think the concrete footings of those baths are still in Brighton now, off the shore. They So this particular white shark was scaring the ladies in the ladies' baths off Brighton by peering in through the bars, and so they decided that they would have to catch that white shark, kill that white shark, and I can't read that from here, but as bait, they fashioned a pig to look as much like a lady as possible. And can you kind of get a picture of that? Can you kind of
Was the pig
get
alive
that
or was it dead?
in
Was it
your
a
mind?
pig
I
body?
assume it was a dead pig. Was it
Okay.
dressed up in a lacy dress? I have no idea. With a bonnet?
That's even weirder.
I don't know. It's very weird.
Yeah.
They caught this shark. They caught a shark off the baths, yep, and brought it ashore. We have one tooth from that shark, which is this tooth here, a white shark, and you can see white sharks have nasty serrated teeth, very nasty serrated cutting teeth, whereas many other sharks just have grabbing teeth. They caught this shark, and when they opened it up, when they opened the stomach up, they found a Newfoundland dog inside also, which had disappeared a couple of days before. So that was a good shark to get rid of. But sharks really are in trouble around the world. The marine food webs need apex predators to keep a balance, and shark numbers are really, really decimated. In fact, there was an article that came out recently that saying that they think that white sharks in South Africa are heading for extinction
Goodness.
because their numbers are down to less than 400 individuals. So sharks are so cool because they can do what we can't. They've got senses that humans can't. They have electrosensory organs in their snout, and they can sense things that we can't. They can sense the electrical impulses in the water of animals nearby, and humans are just really nowhere near as as good as that. So that's why sharks are cool.
Amazing things. So tell us more. You mentioned the basking shark. Now that created a huge amount of excitement last
absolutely.
year. Why was that particular specimen so amazing?
Many museums, basking sharks are known to be in southern Australia. They're more, I guess people interact with them fairly frequently in the northern hemisphere, where they come into large groups of them feed on plankton close to shore. We don't have those plankton schools so much close to shore in Australia, but there are basking shark populations, and there is a suggestion that our basking sharks may be a different species from ones in the northern hemisphere, which would be really, really cool. Yeah, really cool. So they feed on plankton. They feed on krill, the same things that passing whales in southern Australia feed on. This particular shark was caught accidentally by a fisherman working out of Portland, and yes, that's an image of the teeth. That's these teeth, actually, of that basking shark to show how tiny they are. So they feed on plankton, and there's another photo cam of gill rakers, gill filaments or something of that, and like whales have baleen to keep the krill inside the mouth and let the water release.
Looks so similar.
Absolutely.
Yeah.
Basking sharks have the same kind of, they feel a bit like really stiff hair structures, so that they just feed on krill, and this shark was accidentally caught, and we were really pleased that the fishermen decided to donate the shark to science. And it's a difficult thing because that shark was huge, and we have the head, which is absolutely huge currently, and that's the head that was being scanned. So we had the head scanned by a fellow Ben Tan who came down and 3D scanned the head, and so in future, if we make a model of the shark, we'll be able to use the scan of that head and then model the rest of the shark. And so there's
Yes, that's definitely,
so there's the 3D scan, and so there's Ben, a picture of Ben 3D scanning, and then there's actually the scan that is that he's rendered, and then the texture's been put on the outside of that shark. It was fascinating. I mean, he just had a little structure like an iron to go around and scan it, just moving around to scan it, and then he got a wireframe. It had a 3D wireframe that computer software then puts the skin on, renders it, and then puts the texture on the skin. So it's a fantastic way of, we have a little 3D model made of that, and it's a fantastic way of producing giant models of things, because the alternative is to have the animal and put a great big mould over it, like you would making teeth or something like that. Put a great big mould over it and then gradually build it up, whereas this way you can 3D print something in styrofoam.
Yeah.
And then put a skin outside.
Great
so
way
it's
of-
great and a much cheaper way and a much easier way
And
creating
now
models.
we've got that video as a
Yeah.
document.
Absolutely.
I don't imagine you've got some of your specimens there in, is that alcohol or?
It's sitting in 70% alcohol. So these
Yeah.
would have been originally, we take tissue samples and then we preserve them in formalin, 10% formalin, which is essentially embalming fluid. But it's pretty nasty stuff. It's acidic. It will ruin shark teeth. It ruins bone, etches bone. So for long-term storage, we put it in 70% ethanol, so it preserves, in theory, forever.
Yeah, bit hard to do with a whole basking shark though.
Well, we didn't have anywhere to keep it. And so the basking, why it's important is that most museums have bits of basking shark, but they generally just have teeth or bits of gill rakers or a bit of skin because they're so huge.
Great. Now, just a reminder to those of you tuning in, if you do have questions of any of our panellists, please put them forward. We were talking about our collections. Maybe I will ask you a question. Can you guess how many items we have in our collection? And if you can, send in your answers on the chat feed. That'd be great. Any other questions in the chat? Send them to us and we'll see if we can answer them. All right, Eric.
Hello, hello.
Now you come across sharks at times in your
Yes.
work.
Yes, yes.
You coming across these kinds of sharks? How is your work different to what we've been discussing?
No, well, what Di mentioned before about extinction and changes, and everyone here has really talked about diversity, number of species and changes through time and what's happening now, what might happen in future. And I guess I work at the other end, which is what happened a long time ago, and that's the fossil record. So I guess I'm a marine biologist in a way, but I'm mainly a paleontologist, which means I deal with fossils. So they're the remains of extinct animals, and in particular, fossils of things that lived in the sea, namely whales, dolphins, penguins, and sometimes also sharks. And Di had that really nice example of a white shark that was in Port Phillip Bay. And if we think about it, white sharks may not be so abundant today, but they may have been more abundant in the past. And that was just 100, 150 years ago. And then if we think back 500 years ago, we might think, well, were they even more common? Or what was it like 5,000 years ago, 50,000 years ago? And then we get to 5 million years ago. And 5 million years ago, in what's now Port Phillip Bay, there were white sharks, but there were other things. There was a cousin of a white shark, which was as big as the white shark, longer than a Toyota Land Cruiser, but didn't have those nice little serrations, like a steak knife on its teeth. There was also tiger sharks, which today mainly occur in warm waters, tropical waters, so it was much warmer back then. But there was also a shark that had teeth that looked a bit like that white shark tooth, but was the size of my hand. And that was a shark that was the size of the basking shark, but instead of feeding on little krill, it ate other marine animals with backbones, and in particular marine mammals, and amongst marine mammals, whales, including the really big baleen whales that Di mentioned before, that have that hair-like structure growing out of the roof of their mouth. Sounds gross, but that's what happens. Growing out of the roof of their mouth, and they use that to sieve little krill and other hapless little organisms out of the seawater. And one thing that also happened 5 million years ago were whales that we would never have guessed were alive today. So I'm going to show you, this is not a real whale. So this is a model of a sperm whale.
No.
So they're not really this small. So sorry to disappoint you a bit, but think of this as being, think of this as being about 15 metres long. How big is that? Well, if you've ever been on a train, think of a train carriage. And you're getting about that sort of size, proportions, and probably close in weight. So this is a sperm whale, which is alive today, and in fact is often swimming offshore the town of Portland in Western Victoria, where some of you are tuning in from today. And the thing about the sperm whale is that if we only looked at this one species of sperm whale alive today, we would think they're all huge, train carriage-sized, deep diving, and they dive really deep, almost right down as deep as you can go, so thousands and thousands of metres near the seafloor, and they feed mainly on squid, including giant squid. And if that was our only concept of sperm whales, well, that would be it. This is what it would be. But the fossil record tells us a different story. So 5 million years ago, there were sperm whales that had big teeth like this, and were probably not feeding on squid, but instead feeding on other whales. And this is a sperm whale that's probably about the size of that living sperm whale, probably about 12 to 20 metres long. Again, think tram or bus or train carriage sized. And it's a totally extinct species. And its name is Leviathan melvillei. And Leviathan is the Hebrew form of the word Leviathan. And melvillei is named after Herman Melville, who was an American author who in the 19th century wrote the book Moby Dick. Now, Leviathan melvillei is extinct, as I said, and this fossil was found at Beaumaris, not far from where we are right here in Melbourne. It's about 5 to 6 million years old, and it was found just this year by a member of the public like you. Now, I know that some of you are listening in from Portland or tuning in from Portland, and in fact, fossils like this can be found at Portland, along the beach, and if you're really adventurous, and if it's safe to do so, can be found on the seabed in Portland Harbour, including the fossilised teeth of sharks, bigger than the white shark. So these are these amazing discoveries, not only at sea, but also in fossil form on land, that are being made right here in Victoria and nearby Victoria by people working at the museum as scientists. And it just goes to show that we have so much to learn within our own backyard, and we can do that here at the museum, but so can you.
Fantastic. So how does the size of this one compare to a modern sperm whale?
So the tooth of the largest teeth of living sperm whales, this little fella, well, small version of a big fella, basically grow to about this length. So getting up to 2/3, 3/4 of the length of this fossilised tooth here. So
Well,
pretty big, but this is quite a bit larger.
Yeah.
And this is a small one. So teeth of the same species, even larger, have been found in South America.
So why would it need such big teeth? What did it eat?
So-
Were they really sharp at the point, or were they kind of...
They probably
Dull
weren't
like
that
they are
much.
now?
They were probably originally sharp, and they wear down quite quickly, and that's from contact with their food. We know from more complete fossils, so complete skulls of this species of extinct whale that has teeth exactly like this, found in South America, that had very, very strong jaws and huge areas on the skull for the attachment of powerful jaw-closing muscles. And so the thought is that these extinct sperm whales were using these huge teeth to smash and crunch through bone and flesh. So this was really, if Herman Melville thought Moby Dick was bad, this was his worst nightmare.
Wow. So just raw power.
Yeah, possibly. But again, an animal that also had a lifestyle. And there are many aspects. As paleontologists, it's hard for people, for marine biologists studying deep sea organisms, because there's this veil of the deep and dark that prevents that world from being seen, except for very rare and expensive. Often it's quite expensive to go to the deep sea, using remote submarines, etc., and also offshore vessels to actually put nets down and sample. It's even harder for paleontologists. So we can only go so far in estimating, and they really are estimates of what the biology of these extinct animals were like. And that's why I consider myself also a marine biologist, because I have to understand what the living animals are doing in their day-to-day existence, and how many species there are, what they eat, to try and understand what fossil organisms were doing millions and millions of years ago. And that's really hard. And in fact, sometimes it's just impossible. So I will never know what colour this extinct whale was. Was it grey like this, or did it have brown and black stripes? Who knows? It's more likely, though, that its coloration was something like this, and that is dark on top and a bit lighter on the bottom. That's called counter shading, just like the white shark, actually, that you saw in the picture before.
And maybe like the bioluminescent
Absolutely,
deep sea
yeah,
ones.
which
Yep.
are doing it themselves. And tuna
Yep.
and all those open ocean animals
Yep.
generally accounted for. Eric, I have to say, on one of our trips that we did in 2003, in a big expedition out to survey seamounts in the Tasman Sea, we trawled up a tooth of that fossil white shark.
They are there.
And
Yep,
so
they
there's
are there.
many, that species occurred worldwide.
Worldwide,
Yeah.
yep, from pole to pole it
Yes.
seems. But recent studies have shown that they seem to have been more abundant in warmer waters. And indeed, there's fossil evidence of nursery grounds of that white shark. Some of you may have heard of it, called megalodon, in the equatorial region. So, for example, around Panama and also off the coast of Mexico.
Where our white sharks, nursery grounds, the east coast population is around Corner Inlet.
So don't go swimming there? Yeah, maybe
It's not a very nice place to swim anyway, big seagrass beds. Difficult to get into Corner Inlet.
Well,
But yeah.
That's interesting.
Okay, so we've got some questions coming in. Question for Diane. This is from, I don't know who, but it's from Mac. What are the ridges inside the basking shark's mouth?
Basking sharks can, I think I know what you mean. Oh, they're gill openings. So, as I mentioned before, sharks have between 5 and 7 gill openings with gill filaments, which are the structures that take oxygen out of the water for respiration. But they also, so water flows through gills of fishes and sharks so that they can respire. And all fishes, they have gill arches, and fishes, and sharks are fishes, have structures on their gill arches called gill rakers. Fishes that tend to live on other fishes have small gill rakers, but fishes like basking sharks and things like anchovies, which filter feed. So filter feeding fishes have very long gill rakers to keep the food in and strain
Like a sieve.
the water like a sieve, absolutely. So those brownie bits, the brown bits that you can see in that image are the gill rakers, and the kind of bits below them, those long streamy filamenty things, are actually the gill filaments for respiration. So all those ridges are essentially gill arches and the openings between them.
Okay, great. We'll come back to a couple more questions. I would like to go across to Anna now, who, have you completed
I've
your
pretty much
diagram?
finished, yeah. So I was pretty quick actually. I thought it would take me longer today, but
Oh, you're very well practiced.
yes.
So in that diagram, when you're describing the species, what are the features that are going to tell you one species from another?
So yeah, I guess it's completely different for whatever species you're looking at. So for squat lobsters and for a lot of decapod crustaceans, we can look at the body and the spines on the carapace. They're quite useful in that they have lots of different spines, and often the length and the placement of those spines is quite useful for diagnosing a species and distinguishing one from another. So I don't know if you'll be able to see, but these two, this is the one from New Caledonia, so it's pretty far from Australia and from Australia's west coast. It only has one spine here and here, whereas this one has an extra spine on the carapace. So that could be useful in distinguishing these two species, and I'll have to look at more different characters and hopefully find more individuals and look at more individuals of this species to see if that's variable within a species or if that's actually different in all the individuals of this species.
And are you
No, go for it.
also doing DNA work on these? Do you have
We
different
are,
samples?
yeah, so I'm working with someone who's, I give him the tissue and he's doing some of the genetics. So he'll produce, from the work I'm doing, looking at Australian species, we're trying to work out how all those different species of squat lobster are all related to each other, and that can help to try and determine whether these two species are different. So luckily this New Caledonian species does have, we've got data on its genetics and we'll be able to compare that and tell. But it's useful to have both the genetics and the morphological data as well, because Sometimes there's species that look different morphologically but are actually the same, and vice versa.
Yeah.
So, yeah.
It's one of the problems with deep sea work in terms of these days we do a lot of genetics, and it's hard to get animals from the deep sea. And we've got lots of specimens from before 70s and way back, but we've had them fixed in formalin, which is difficult to get DNA from formalin's fixed material. So in a sense, we're collecting again
So for the first time.
for the first time to
good
get
DNA
for
material.
DNA.
We might have time for one or two more questions. What's the strangest organism you've discovered? Anyone found something that was just...
Ask Hugh.
Yeah.
I reckon we have a
Cute.
pretty weird animal. You want to put a photo of the dendrogramma up there? Just as a, so this is about 20 or 30 years ago. Some scientists were exploring the deep sea in around the around Tasmania, and they found these really strange rubbery mushroom things. So can you zoom out a little again, Ken, to give us a full shape of the thing? So these really strange creatures, and they're maybe about a centimeter wide, and it looks a bit like a mushroom. It has a stalk, and it has a kind of a cap, but they're transparent, and as you can see if you look really closely on them, they have these really weird branching lines, and they're so unusual that it took at least almost 30 years before anyone could actually identify what they were. And even then they said, we're going to give it a name, and they called them Dendrogramma enigmatica. And dendrogramma means like a dendrogram, which is like a tree diagram, and because the branching lines on the disc look kind of like these branching diagrams. And enigmatica, the species name, because it was enigmatic, because they still didn't know what it was. So they gave it a name, but they couldn't place it anywhere on the animal tree. They said it's not a jellyfish, it's not another type of like coral or something, and a bit like Di was saying, because the way it was collected at the time, we couldn't do any genetic tests. We couldn't do any DNA, so we couldn't figure out where it belongs. It was just based on the shape and the look of it. But last year, when we were out sampling in the deep sea in South Australia, we were really lucky. We found these things again for the first time in 30 years. So we were very, very excited. So we managed to get some DNA samples from that. And we did the analysis, and that's actually been able to show exactly what these dendrogramma are. And the dendrogramma, turns out, they are a type of jellyfish. They belong to a type of jellyfish that's a little like a bluebottle. They're called siphonophores. Candace, do you have a photo of, I believe, what one of them looks like? I think you've got maybe not. I don't think I did. Oh well, never mind. I thought I had a photo of the whole thing. But these things were incredibly strange. And as it turns out, they're not whole living animals. They're actually a piece of a larger animal that sort of fell off when it was being collected. So we found these things. They're so strange. They're not even the whole animal themselves. So the real animal would look like even kind of a small, floofy little thing with some little tentacles on it.
Like a Pokemon.
Yeah. Well, no, I don't know any Pokemon that look like that.
Okay. Maybe not.
So it looks like a little orange, kind of floofy little kind of ball, but then it has these little mushrooms that come off the side of it. So it's one of these things that we found the little mushroom piece, but not the rest of the animal. And so when we were left with just that, we had to try and figure out, what is this? And so that's one of the real mysteries. And it just goes to show this stuff out there that's still completely unknown. And when you do come across it, you can it's like looking at it for the first time. It says, well, I have no idea what this is. It's always a surprise when things come up.
I think there's more stuff out there that we don't know in the
Absolutely,
deep sea.
absolutely. I
much
think
more.
we only know a tiny amount of what's out there.
And as you said, it costs one heck of a lot of money to get
It
out
does.
there
It does.
and ship time.
Now we're having some questions about what you have studied to get there. So give us the 10-word answer. What sort of did you do?
Science. So a science degree in undergrad at university. So that was about 3 years of study. Then another year of study for what's called an honours year, where you do a small research project. And then another 4 years of study, or what's called a PhD, or a Doctor of Philosophy, which is a much longer research project. That's in my case. And then, if you like, some additional study of a year or two. So if you're really interested in pursuing a career in science and discovery and being a scientist, at least the kind of scientist that we are, it can involve a lot more study after school, so be prepared for that.
And we'll had similar kind of pathways.
Yes, yes.
Well, yeah, I did a master's degree in fish. I was lucky enough to get a job. I actually had an paid internship at the Australian Museum years ago, things that they don't fund anymore.
Outrageous.
Lucky.
6 weeks worth of paid money to work on a project straight out of university, and I found my home. And while I was there, I did a master's degree. But I would say follow your passion.
Yeah, absolutely.
Absolutely follow your passion.
But I guess I'd say I didn't always want to be a marine biologist, but I did like animals and I liked biology. So at that time I just went into uni and took lots of different subjects, and then marine biology was just really cool. And I started working on fish in seagrass, and then just ended up in working on squat lobsters in the deep sea. So
One thing I'll mention real quickly is that to all of you out there, if you're interested in biology or even in marine science, it doesn't have to be just studying in your, the biology class. Biology is an amazing field that you can do a whole bunch of different things and still wind up working. If you really like programming, like that's, you can be a programmer and do, write software or analysis that we're completely reliant on people, to do things like that. If you're really interested in genetics or chemistry or ecology, so you can do biology that's really maths heavy. You can do biology that has almost no maths at all. You can do stuff that's really chemistry heavy or not. It's a job or a field that can handle a whole variety of different skills. So if you're passionate about sort of marine science, you can really come at that from a whole bunch of different angles.
Great. We're going to have to wrap it up. Thank you very much for joining us, and thank you for joining us too.
Thank you.
Before we go, we would like to say that if you've enjoyed this, we do regular live streams. We also have some events coming up in National Science Week, which is in about 3 weeks' time, called Meet the Scientists. You can come to the museum, meet Eric again, and a whole range of scientists from all different disciplines in the museum. Just check our website for details. So thank you all very much.
Thanks, Elka.
Thank you.
Thanks,
And
Olga,
thank
and
you.
thank you.